Hello friends, myself Dr. Smily Pruthi. I'm national level faculty teaching biochemistry. In this video, I'm going to discuss November AIIMS 2017 biochemistry questions which came in your exam yesterday only. So let's get started. The first question which I'm going to discuss is a 10-year-old boy rapidly develops hypoglycemia after moderate activity. Blood examination reveals raised level of ketone bodies, lactic acid and triglycerides. On examination, liver and kidneys are enlarged. Histopath shows increased glycogen deposits in the liver. What's the diagnosis? This answer is von Gierke's disease. Von Gierke's disease is the most common glycogen storage disease. This question has given you the typical clinical features of von Gierke's disease. In von Gierke's disease, we have severe hypoglycemia, ketosis, enlarged liver and kidneys, hypertriglyceridemia, lactic acidosis, and hyperuricemia. Hyperuricemia, why it occurs? Because of increased lactic acid. Because lactic acid competes with uric acid for excretion by kidneys. If we have lots of lactic acid, then kidneys will keep excreting lactic acid, leaving uric acid in body, leading to hyperuricemia. What is the enzyme deficient in von Gierke's disease? The enzyme deficient is glucose-6-phosphatase deficiency. This enzyme converts glucose-6-phosphate to glucose. So when this step is not occurring, then the product glucose not formed and the substrate glucose-6-phosphate accumulates. Accumulation of this glucose-6-phosphate occurs in the liver, which will lead to increased glycogen deposits. That's why hepatomegaly occurs in these patients. And this glucose-6-phosphatase enzyme is a common enzyme to two pathways. That is glycogenolysis, also for gluconeogenesis. So both these pathways are unable to give glucose in the blood, that is glycogen also and gluconeogenesis also. So these patients have severe hypoglycemia, and whenever we have severe hypoglycemia, then this leads to ketosis. And why increased triglycerides will occur in this patient? Because excess of glucose-6-phosphate will be converted to pyruvate also, which will form lots of acetyl-CoA, and this lots of acetyl-CoA gets converted to fat in the liver, and that is mainly triglycerides, right? So hypertriglyceridemia also occurs in these patients. So let's quickly look at all the glycogen storage diseases here. Type 1 is von Gierke's disease. I told you the enzyme deficient is glucose-6-phosphatase. It is the most common glycogen storage disease. Organ affected is only liver. Then type 2, Pompe's disease, affect lysosomes. Only type 2 glycogen storage disease affect lysosomes, and any other disease of glycogen will affect which organelle? Yes, cytoplasm, right? Now, in Pompe's disease, the enzyme deficient is acid maltase. The next one is type 3, Cori's disease or limit dextrinosis. The enzyme deficient is debranching enzyme. Cori's disease, debranching enzyme. Easy to learn. C for Cori's, D for debranching, CD. Then Anderson, A for Anderson, B for branching enzyme. So A, B, C, D. It's easy to learn like this. Anderson disease, also known as amylopectinosis. Cori's disease, also known as limit dextrinosis. Now these three diseases, type 2, 3, 4, they affect all the organs wherever glycogen is stored, that is liver also and muscle also. And some amount of glycogen is stored in brain also, so brain is also affected in these three diseases. I'll give you one trick for these question solving. If they give you this clinical picture that liver, muscle and brain all are affected, then you're very sure either it is type 2 or type 3 or type 4. Now in this clinical picture, if they write lysosomes are affected, then you're very sure it is Pompe's disease. If in this same clinical picture, instead of lysosomes, they write limit dextrins are increased, then you're very sure it is type 3 Cori's disease. And if in this clinical picture they write amylopectins are increased, then you're very sure it is type 4 Anderson disease, right? So easy to solve these questions. Then type 5 is McArdle's disease. In McArdle's disease, the enzyme deficient is muscle phosphorylase. In Hirsch's disease, enzyme deficient is liver phosphorylase. So respectively, the organ affected will be muscle in type 5 and liver in type 6. Now, one very good confirmatory diagnostic point for type 5 McArdle's disease is lactate normal after exercise. Lactate normal after exercise. In a normal person, like if you and me, if we will do exercise, then our blood lactate levels will be increased, right? Because of Cori cycle. But if this patient will do exercise, then lactate levels will be not increased because the very first enzyme to break down glycogen in muscles, that is muscle phosphorylase, is not there. So glycogen not broken down, glucose not released, so lactate not formed, right? Now, the next one is one thing important I would like to tell you here. In Warnberg's disease also only liver is affected. In Hirsch's disease also only liver is affected. So there are few points by which you can distinguish between them. In type 1 disease, there is severe hypoglycemia, but in type 6 disease, we have mild hypoglycemia because in type 1, no gluconeogenesis is occurring because this enzyme, glucose-6-phosphatase, is common enzyme to both gluconeogenesis and glycogenolysis. That's why severe hypoglycemia in type 1, that's why ketosis occurs in type 1. Ketosis does not occur in type 6 patients. Right? So the next question is, which amino acid does not include post-translational modification? Examples are selenocysteine, triiodothyronine, hydroxy-proline, hydroxy-lysine. Answer to this question is selenocysteine, which is the 21st amino acid. So the 21st amino acid, selenocysteine, is given by codon UGA. 22nd amino acid is given by codon UAG, that is pyrrolysine. Both these, they are stop codons, but by some modification, usually stop codon do not give rise to any amino acid, but these two amino acids, these two codons, by some modification, they can give rise to these two amino acids, selenocysteine and pyrrolysine. And this modification is not post-translational modification. This is co-translational modification. That is, during the translation, these changes are occurring, right? Now, one more important point I can include here is that enzymes which use selenocysteine, the 21st amino acid, at their active site, they're known as selenoproteins. Mostly these selenoproteins are peroxidases and reductases. I can give you a few examples which are very important, frequently asked question. That is glutathione peroxidase, thioredoxin reductase, and iodothyronine deiodinase. These three enzymes require selenocysteine. They are known as selenoproteins. Next question, glucosamines used in following condition. Options are arthritis, Niemann-Pick disease, Alzheimer's disease, cancer. So answer is arthritis. Glucosamine is an amino sugar, and amino sugar is important constituent of GAG. GAGs are made up of tandem repeats of amino sugar plus uronic acid. Amino sugar plus uronic acid. And GAGs, they combine with proteins. GAGs, these linear chains of amino sugar and uronic acid combine with proteins, making proteoglycans. A very important proteoglycan present in cartilage is agrican. Agrican is the major proteoglycan in cartilage. So it requires this glucosamine, and therefore this is a very common thing that people are using glucosamine along with chondroitin sulfate for arthritis, for relieving the pain in these patients, and for improving the joint functions, right? Now, next question number 4. Which vitamin is required for glycogen phosphorylase enzyme? This is the rate limiting enzyme for glycogenolysis, and it requires PLP, that is pyridoxal phosphate which is vitamin B6. This PLP for this enzyme is not required as a coenzyme, but it is required as a phosphate donor. Because this enzyme, glycogen phosphorylase, will cut glycogen. Alpha 1,4 bonds are cut and the glucose released are transferred inorganic phosphate. So that phosphate is taken from PLP, right? Next question, question number 5. Structure of DNA is shown. This was an image-based question asked in the exam. Structure of DNA is shown, and they have asked that what are the bonds present in between two nitrogenous bases. You know, between A and T we have double bond. Between C and G we have triple bond. These bonds are hydrogen bonds. So answer to this question is hydrogen bonds. Let me tell you all the bonds between in the structure of DNA. The DNA structure is double-stranded. There are two strands. So if these are the two strands, then the strand backbone is made up of sugar and phosphate. Sugar-phosphate backbone. And nitrogenous base is attached to sugar. In between two nitrogenous bases, we have double bond or triple bond, and you know this is hydrogen bond. In between nitrogenous base and sugar, this is beta N glycosidic bond. In between sugar and phosphate, we have 3-5- phosphodiester bond. This beta N glycosidic bond and 3-5- phosphodiester bond, both these are covalent bonds, that is strong bonds. So suppose these are two strands of DNA. The two strands are joined by hydrogen bonds, which are weak bonds. So it is very easy to break all these strands, to break all these bonds, and we can separate the strands. We can very easily separate the two DNA strands. But each strand is having all the covalent bonds in it, which are strong bonds. So it is very difficult to break the individual strand, right? Next question number 6. Following are the tests done for protein, sugar and ketone bodies, which will be positive in starvation state in urine, right? So these are all the tests done in urine of the patient, and what will be positive in starvation. You know, ketone bodies are positive in starvation, but now you have to recognize the test tubes also. The first one is biuret test, which is done for proteins. Next is Benedict's test, which is done for reducing sugars. Then next is this ring shown. It is Rothera's test. A purple colored ring is formed at the top. This is very typical of Rothera's test. And this patient will be having ketone bodies in starvation in the urine. So answer will be only 3. Option C, only 3 in third number test tube, right? Here the numbers are also written 1, 2 and 3 number. Then you can mark 3 here, right? For proteins, we have biuret test, which is only positive if 2 peptide bonds are present in the protein. That is, a tripeptide will be showing biuret test positive. Then for carbohydrates, the most common test which is performed is Benedict's test in urine, which is positive for reducing sugars. And for ketone bodies, we have seen the Rothera's test, purple colored ring, which is positive for ketone bodies. One more test for ketone bodies is Gerhard's test. Both these tests are for ketone bodies, Rothera's and Gerhard's test. Next question number 7, which is not involved in iron metabolism. Options are transthyretin, ceruloplasmin, hepcidin and ferritin. Answer is transthyretin. Let's see the detail. For iron storage, we need 2 proteins, ferritin and hemosiderin. The difference between ferritin and hemosiderin is that hemosiderin contains more iron content as compared to ferritin, or you can say so many complexes of ferritin are formed with less of protein content and more of iron content, and that is hemosiderin. Next, for iron transport, we have transferrin, not transthyretin. For iron storage, we have transferrin. Then ceruloplasmin is a copper containing enzyme which is required in many many oxidases. Copper is required for many oxidases, and especially here ceruloplasmin, which is copper containing enzyme, is required for ferrooxidase activity. This ferrooxidase can convert Fe+2 to Fe+3, that is ferrous to ferric conversion, and this ferric form of iron is very much required as a transport form of iron, right? Then hepcidin is a protein which is helping in the regulation of iron transport in the circulation, and transthyretin is the answer, which is not for iron metabolism. It is required for the transport of thyroxine and retinol binding protein. So these two terms are confusing, transferrin and transthyretin. Transferrin is for iron transport, transthyretin is for thyroxine and retinol binding protein transport. See the name transthyretin. Retin, see the name like this, trans for thyroid and retin. Thyroxine and retin is retinol binding protein. So the name is telling you. Earlier, this transthyretin was named pre-albumin because it was migrating faster than albumin on electrophoretic gel. But now the name is transthyretin, so it is for the transport of thyroxine and retinol binding protein. Next question, in SCID, that is severe combined immunodeficiency, which type of DNA repair is defective? And the answer is end joining repair is defective. So you can see all the defects which are commonly asked. Defect in non-homologous end joining repair is SCID, severe combined immunodeficiency. If defect in homologous repair occur, then various syndromes are there. Bloom syndrome, Werner syndrome, breast cancer, also increased susceptibility to breast cancer can occur. Then nucleotide excision repair defect will lead to xeroderma pigmentosa, and mismatch repair defect will lead to HNPCC, that is hereditary non-polyposis colorectal cancer. Question number 9. Child presents with hypotonia and seizures. It was confirmed that this child is having Zellweger syndrome. Which of the following accumulates in brain? Answer is long chain fatty acids accumulate in brain in this patient of Zellweger syndrome. So let's see the detail. Zellweger syndrome is also known as cerebro-hepatorenal syndrome. So what occurs in brain is that hypomyelination occurs, impaired neuronal migration occurs, so very severe kind of neuronal damage occurs to these patients, and they have a problem in liver, that is hepatomegaly, and usually these patients have renal cyst found in them, so that's what is known as cerebro-hepatorenal syndrome. This is a rare autosomal recessive disorder. There are absence of peroxisomes in almost all the tissues. Peroxisomes are responsible for the oxidation of very long chain fatty acids, that is which contain more than 22 carbons. So accumulation of polyenoic fatty acids having more than 22 carbon occurs in brain, and severe neurological symptoms are there in the patients, and most patients die within first year of life, and this is a rare disorder. Last question, question number 10. Dried blood drop of an infant can be used to know. Answer is inborn errors of metabolism. This technique has been used since last 8-10 years. You must have heard of Guthrie's card. Guthrie's card are for phenylketonuria. And what is done is that a blood spot from the heel of the newborn is taken, and it is taken on the filter paper, and then the filter paper is allowed to air dry for some time, and these samples can be preserved for years. So what are the advantages? That we require minimal volume, minimal training is required for the staff who's handling these samples, ease of transport and sample stability for so many years, and once dried, the sample is stable for months to years. Various inborn errors of metabolism which can be detected by these dried blood samples are phenylketonuria, galactosemia, maple syrup urine disease, which is defect in the catabolism of branch chain amino acids, valine, leucine, isoleucine. Then MCAD deficiency, that is medium chain acyl-CoA dehydrogenase deficiency. Sickle cell disease, these kind of disorders can be detected. So we're done with all the questions. Thanks for watching and best wishes for your future exams.